is Rob Schuler. He's going to take us through an AI-ready platform for biomedical research in FaceBase.
Thank you. Yeah, so I'll be talking about really what FaceBase is doing to enable AI readiness for dental, oral, craniofacial related research as an end-to-end platform to support that. So, clearly there are tremendous opportunities for AI in biomedicine, whether it's examples like classification of skin lesions for better diagnostics or cardiovascular risk scoring for predictive outcomes, or pattern recognition and genomics for just broader precision medicine discovery. Trying my time. But all of this really hinges on whether it's built on solid data foundations. And that's really the key to enabling these exciting opportunities. So, and it doesn't take going back too far to really see a cautionary tale here. So during the COVID pandemic, there were a lot of models that were developed, whether for diagnosis or prognosis during that time. And unfortunately, the retrospective analysis of them is that they really didn't pan out in terms of providing really any benefit for clinical use. And there were a couple of interesting studies, one that looked at over 600 models for COVID prognosis, another one, 62 papers for COVID diagnosis or prognosis, and they all really fell flat. And the issues all came down to matters of data. The root causes were things like inadequate sample sizes. This can lead to bias risks that were unaccounted for in the study design, so-called Frankenstein datasets that are stitched together from different sources, but actually end up having similar patient data, the same patient data in them, and therefore resulting in models being tested on data that they were actually trained on, and many other factors. There are interesting papers here that I reference for, you want to dive into those. And unfortunately, the reproducibility crisis really extends really beyond COVID and beyond just even ML-based science. So an earlier survey asked scientists across a variety of domains whether they could reproduce each other's work, and about 70% said that they had experiences that failed to reproduce others' work, and a majority agreed that there was a significant concern for reproducibility in general. Another survey looked at over 329 papers for machine learning across 17 different fields. So this wasn't just specific to biomedicine, but looked across different fields and found that there were various issues with what's called data leakage across them. Data leakage is basically there are some kind of either samples that show up in testing or validation sets, and there were subtle issues too, like let's say you have a longitudinal study. Well, you might have multiple images for the same patient. Those are two separate images, and in some sense, they could be in your training and test set, but because they're from the same patient, they're actually highly correlated. And so just not enough independence between them. And so again, finding significant methodological issues in many papers. And that report is not that old. That was 2022. So where we come in and what we'd like to say is that AI ready data, there's talk of AI ready data and what is it, how should it be annotated? How should it be curated? And this work has kind of gone on somewhat independent of the overall FAIR data in the biomedicine. Our observation is that AI ready is really mostly just FAIR data with additional context for experimental context and provenance over time. First of all, we would argue that this requires a community effort. It's not an individual effort. If you look at major discoveries like AlphaFold, major advances like that, it really took tremendous amount of data collected by entire communities. It wasn't just one lab's data that went into that. Of course, in the AlphaFold example, that was built on the protein data bank and the many decades of careful curation of data that went into that. The next thing is that our objective with AI readiness is not simply to be able to rerun an experiment, but we actually want real reproducibility of high quality, robust results. And so it's not just simply a matter of having the scripts and being able to rerun it. We want to know very precisely what data are those scripts run on. I don't know if you've looked at any papers that report on AI results or AI models that have been developed, but one recent one I was looking at, for example, basically said it was a work on classifying mouse skeleton, which will be relevant to a later part of my talk. And when it came to the data availability statement, it basically said the data for this was available, was collected from IMPC, and it's available there. It doesn't really tell you what files, nothing about the checksums of them, no way to know exactly what data was actually used in their experiments. And that's kind of endemic. And then finally, as I said, we observed that FAIR data really covers most of what one talks about in terms of AI readiness. And I think beyond that, you have to question whether, do we really want to share pre-processed features? I think probably not. We want to share carefully annotated data and let scientists then build their features from that. So one of the things that we talk about are the scale principles. And I won't go into this in too much depth, but just as background, we argue for data repositories to support the kind of community or the kind of critical mass of data to, first of all, be built on the principle of self-service curation. You've heard that mentioned already, but this means that we want to empower the entire community to be able to curate high quality data, and so that we can achieve the kind of scale of data needed, the kind of critical mass of data needed to support AI research. Secondly, those platforms need to be domain agnostic. As you've seen, like we're dealing with data from across genomics and imaging, and not just craniofacial development, but dental and public health and related data such as the inner ear database. And we need to be able to support a broad range of research data. The next thing is that we argue that you need lightweight information models, that we need enough detail to support those FAIR principles, interoperable, reusable, but this kind of goes hand in hand with a self-service curation. You don't want to overburden our community with too highly of detail of metadata to be able to curate into the system. And then finally, evolvable systems. So the systems need to be able to adapt as we go forward. I mentioned how we are now supporting more highly detailed information on patients and visits, such as part of the craniorate, craniosynostosis data, as part of the ear-based database. And all of that has been done through really rapid evolution of the system, and that's built on the underlying principles of our system. So the interesting thing is that when you come down to it, this really enables agents, AI agents, to be able to work on the FaceBase platform. And this comes down to open, well-documented interfaces. So all of the underlying standards, those are based on things like the HTTP protocol. That's one of the underlying protocols of the web. And by having things that are really standard-based, open interfaces, and well-documented, we find that agents work very well over that. The next thing is that, of course, FAIR data then leads to machine interpretable representations of the data. And then finally, our system is based on a formal meta model exposed through that underlying platform. And this is based on something called relational data model. I won't get into the technical details of that. But the interesting thing is that when you look at the kind of language models that are being developed, they're being developed to be able to program. They're being developed to be able to understand relational data. One of their targets for training, their benchmarks, are these database targets. And we actually expose those through our platforms, and it provides a really unique environment for agents to be able to operate over FaceBase data, as I'll demonstrate in a moment. So when we look at what the agent's role, we see this as an overall FaceBase role, that we're not just a place to dump data in and then come and extract and go do your own thing. But actually, there's an end-to-end life cycle here that involves FaceBase that we can enhance with the AI agents that we're developing. First of all, it begins with discovery, just being able to translate your intent to find certain data into detailed queries over the platform, then organizing and summarizing data throughout it, assembling those data sets into something you can work on, extracting them, staging and extracting them to download to your own platforms, and then bootstrapping through an initial script to get you started. Slowly materialize it there. Okay, so agents can work throughout this entire life cycle. So just to demonstrate here, I've got our newly released agent, and you can actually go to the FaceBase site and try this out yourself. But let's just begin with the first step here, asking the broad query. I'm going to try to read my own typing here. Okay, so tell me about craniosynostosis and related data in FaceBase. So you can just ask a broad question like that. It does a little bit of thinking there. You might notice it's a little hard to read, but at the top there it says it used one tool. Now, what that is that we're providing specific interfaces for agents to work with FaceBase. So this is a little different than just going to a public, Gemini or ChatGPT or other chatbots, but we're actually giving it tools that it can do a deeper search into FaceBase. So this comes back with some general information about craniosynostosis, and then points out a few of the data sets and asks if you'd like to look further into it. So the next part of that step is really expanding and going a next level deeper. And so we want to kind of, just as an interesting thing here, just challenge it a bit. So yes, get details on craniosynostosis, related data sets, also summarize data sets that share common phenotypes. Come on, Rob, type faster. Okay, phenotypes with craniosynostosis. All right, so that's kind of an interesting query because that's not something that you can really directly go and just facet through. It's asking the agent, it's going to have to think a little harder this time, but it's asking the agent to not only find things that are directly related to craniosynostosis, but find things that have some related some sort of overlapping phenotypes with it, maybe broaden out what you might be able to find. It's thinking harder, it's working harder, use 9 tools. Okay, you don't need to know exactly what those tools are, but basically it has structured queries that it could do within FaceBase to find more related information. And now it starts spitting out some things. Okay, primary craniosynostosis data sets here, giving you the full details right away. And it's just pulling this information straight out of FaceBase. Then data sets with shared phenotypes. It talks about a few things. There's some abnormal facial spacing. I saw something about sutures in there. So it looks like it's picked up things that are relevant. That's just a query that, while you could do it through the usual faceted search paradigm, it would be a bit more challenging. And then finally drill into the data and get us started. So this is another kind of fun one here. All right, so now generate a script for me to fetch and get started on analysis of the relevant data sets. Okay, so I'm a bit loose there. I could say specifically which data sets I want, but I'm just letting it decide for me at this moment, which, normally I would be a lot more specific. And then it starts generating scripts. So this capability, and then also I should mention this capability is really enabled by the fact that we have well-defined APIs. We release those in, the Python. They're also usable through R. Documentation is very precise on our side, both for the underlying protocols as well as the programmatic APIs. And so, agents are really trained on the ability to program, to query databases. So they're with a well-documented underlying platform, it can really do a lot of great things that it wouldn't have otherwise been able to. Everything on the FaceBase site that you see in the GUI, in the user interface, through the website, it's all available through the underlying APIs and the interfaces. So that's kind of a really, I hope you enjoyed that. It's really pretty remarkable what the agents can do when you give them enough information. So the next thing is, so we've been able to find the things that we wanted to grab. So through the FaceBase catalog, you're able to get very clear definitions of your data in machine interpretable formats. You're able to extract them with persistent identifiers and in a archive format called the big data bag, which gives you very precise references to all the data that you're going to use in a training set. And then we have an experiment management framework that is under development called Deriva ML. And this extends from our platform to helping you with the actual machine learning experiments. And just to look at what this is like under the covers, this brings together a domain-specific schema. So in the colored boxes here are the face-based domain concepts with some reusable machine learning model elements that track the machine learning data sets and workflow execution and so forth. So you can track all of your provenance. It allows you to compare between experiments, the protocol, the parameters that you used on them, and really trace back any failures of those experiments and figure out what's going on. So the agent's role in all of this is to accelerate the experimental loop. It can help with configuring and running the experiments themselves, to enforcing best practices. So a lot of times, those best practices I mentioned up front, a lot of those errors were basically the COVID and other errors are because not following very rigorous methodological approaches. So we can help remedy that through agents that support those, that can basically codify those as agent skills. And then finally, agents can take care of the meticulous bookkeeping. So you want to track all the results very carefully and the parameters. The agent framework can support that. So let me just give a little example here. This was a model that we put together. And we know that we have these highly curated collections of images. And in this particular case, we have the database within FaceBase called MusMorph, which has normalized images for about 10,000 different samples. And we wanted to ask the question, could we train a model just to be able to tell the difference between the mutants and the controls, the normals in this? And then secondly, would we be able to train them? If we could do that, could we train the model to detect which of the gene labels belongs with those images? So these images are labeled with a particular gene that was in some way mutated. It could be a heterozygous or homozygous mutant. And so we posed that question. We put together a fairly sophisticated extension of the residual network convolutional neural network architecture extended for 3D images, and so we trained it. Sorry, I'll go back here. We trained it on a careful stratified sampling of the images, careful to separate out our training from our validation and the held out test, and then our result. Drum roll, please. Okay, so our result here. So our first model, mutant versus control, proved to be have very good predictive accuracy in terms of being able to correctly label. This was the control versus the mutant at about 85%, and a number of other measures there that we can look at were all pretty satisfying. I think the biggest limitation is that it's a little better at detecting mutant versus control, but still the control detection prediction is still pretty good. So then we thought, well, the really harder one is going to be, can we tell which gene we should, label belongs on these images? Because those have to be very subtle. And remember, this is not using landmarks. This is just looking at the volumetric information. So it has nothing but just the micro CT volume data. And all right, I didn't get a drum roll, but okay. So on the second one, we also got a very surprisingly good predictive accuracy. So the top one accuracy was again in the 85% range. And the interesting thing here is that the top three accuracy is very high. So when it's wrong, it's not wrong by much. So the top three accuracy was 97%. And another interesting thing we found when we looked at the errors on this second model is that, and I'm getting a little bit out of my depth here, so don't beat me up too much, but what I can tell you for sure, okay, is that we can look at we have some confusion graphs over here. Is that the errors aren't just random all over the place. That the errors are potentially explainable. And again, that's where I'm getting out of my depth here. But if we look at it, pretty much the images that were labeled with, say, Sprouty 1, 2, or 4 are just confused for each other. They're not confused for some random gene out there. The HDAC9 is tightly related, symmetrically related, or sorry, asymmetrically confused for twist1. ISL1 and MSX are pretty much symmetrically confused. And there seems to be, and again, I'm getting out of my depth there, but there seems to be evidence that those are related, but they're not just random errors. So it's probably not an artifact of just picking up some confounders from the scanner or site effects or something like that. So that's really interesting, and it makes you wonder if we had thousands of more gene labels on thousands more images, what other interesting correlations might we find? These are all probably are known connections here, but imagine if you were finding other statistically strong indicators that there was phenotypic expression for different genes that you didn't expect, and you could dive into that further. So the opportunities are quite interesting. All right, so okay, so I'm concluding right there. So overall, this work and these models are really an artifact of the overall infrastructure underneath it. Robust findability, version splits, tracked execution, and analyzable error structure. So with that, I just want to just make the point that FaceBase is really pushing towards supporting AI readiness end-to-end in the research lifecycle, and not just a place to deposit and extract data. And with that, thank you.
Questions? Yes, Brendan.
A comment and a question. The comment, it's interesting. You pointed out as errors, and they're explainable. That, from an experimental perspective, very powerful. Many times in development biology, we're looking for phenocopies to help point to genes within the network downstream. So I do think that that's a very valuable element. It's the flip side, sort of the cup half full side. But relevant to that, I wasn't clear. Have you tried to benchmark that with external data? Or is that, I wasn't clear on how you present it. Like, could I send you a micro CT of skull? And would it be able to?
Right. So at this point, no. This is all within the, this is kind of like an initial attempt, all within the MusMorph collection of data, which have all been curated through processed through a common pipeline. That way, kind of remove some of the variability. I think we will be having a talk tomorrow where they've used both MusMorph and other data. So that is kind of a next step to then apply this and see how well it does.
That would be the and you would incentivize investigators to actually contribute.
Absolutely.
Because they're getting something in return.
Yeah, absolutely. Yeah, thank you.
I would comment as a general direction, one of the reasons why we're pushing on this recording the artifacts and this notion of reproducibility is not taking the same code and running it and getting the same answer. So this whole thing with version data sets, provenance on execution, very careful code management, looking at it, and it now gives you a framework, reusable features, right? It now gives you a framework to do real comparative analysis, right? I have the exact same data, right? I've rerun this. Now treating that as experimental regime. Well, how sensitive is this to the learning rate? How sensitive is it to, this particular data set I used as an input? So now we have a controlled environment that allows us to benchmark and see whether we actually have a real result or just not a fluke artifact.
Relevant to your first example, your output were the files that were needed for the next step. Could it do the compute within FaceBase, or does it have to be downloaded locally?
We that Deriva ML framework is it is a fully what's called containerized environment, and you actually could deploy that to the cloud and run it. I wouldn't necessarily say in FaceBase because then we'd be paying for the compute, the GPU and stuff like that, But the environment is containerized where that absolutely it can run on your laptop. It can run in a cloud
You
environment
still have to egress to another part.
or at least to another part of like the Amazon cloud where we run. And so run that there. And then that wouldn't necessarily be an egress charge, but would be just to another compute node on the Amazon framework. So one could do it that way.
We are running into issues with different people who use Amazon. There's
Sure.
So one thing we can do, because of the way FaceBase is structured, that you can leave, if you have the right cloud setup, right? You could leave the data deposited, right? Move the management environment to your own platform, your own payment, reuse those S3 buckets, but still have all the reliability. So because we're still managing then those collections of files in a very rigorous way. So we can pick the whole thing up, move it in a very lightweight environment, and if you can compute in place, you can.
Okay, I think that's all the time we have for that. Thank you so much, Rob.